Hey everyone, and welcome to The Explainer. Today, we're going on a journey that's honestly going to completely change how you look at the world around you, and yeah, even the way you look at yourself. We are diving deep into the awe-inspiring science of microbiology. We're going to map the microscopic universe, exploring these incredible, invisible forces that literally shape our planet, our environment, and our own bodies. It's this massive hidden world, and understanding it is basically understanding the very foundation of life itself.
To really put this in perspective, let's start with a huge question. What ruled the Earth 3.8 billion years ago? So, our planet is about 4.5 billion years old, right? But the oldest fossilized rocks, which are called stromatolites, show us that bacterial life was already thriving 3.8 billion years ago. Early Earth had a completely anaerobic atmosphere, literally no oxygen at all. But then came cyanobacteria. These little microscopic pioneers evolved the ability to use water for photosynthesis, and they just started pumping out oxygen gas as a waste product. They single-handedly oxygenated our entire planet over billions of years. All present-day life evolved from these ancient bacteria. Pretty wild, right?
Okay, so here's our roadmap for today's explainer. First, the invisible world discovered. Second, sorting the microbial chaos. Third, the three domains of life. Fourth, bacteria, the ancient survivors. Fifth, fungi, viruses, and beyond. And finally, number 6, our microscopic co-inhabitants.
Let's jump right into Section 1, the invisible world discovered.
Before we get into the history, I've got to hit you with a totally mind-blowing fact. 10 to the 14th power. That is 100 trillion. A single human being contains roughly 10 to the 13th power of their own human cells, but you harbor 10 to the 14th power of bacterial cells. In fact, 10% of your total dry body weight is entirely bacterial. You are, quite literally, a walking, talking ecosystem.
So how did we even find out about our tiny tenants? Well, it took technology. Back in 1674, an amateur microscope builder named Antony von Leeuwenhoek first observed and sketched out what he called animalcules. But microbiology didn't really explode as a true science until the Golden Age, right between 1870 and 1920. That's when heavyweights like Louis Pasteur and Robert Koch totally demolished the old idea of spontaneous generation. They established the germ theory of infectious diseases, proving for the first time that specific microbes cause specific diseases like anthrax and tuberculosis. And this foundation just grew rapidly from there. By 1940, Selman Waxman discovered vital antibiotics like streptomycin, and then in 1953, the massive mapping of DNA by Watson, Crick, and their team cracked open the era of modern molecular microbiology.
Moving on to Section 2, sorting the microbial chaos.
Once we realized there were countless invisible life forms out there, scientists faced a really massive challenge. How on earth do you actually organize them all? Now, what's super interesting here is the evolution of how we categorize life. Historically, we relied entirely on phenotypic characteristics. That means grouping microbes by things we can actually see, observable traits. Like what shape are they? Can they move? How do they react to specific dyes, like the Gram stain? It's kind of like classifying animals by whether they have fur or feathers, right? But modern science has totally shifted to genetic and epigenetic analysis. Now we classify microbes by mapping their actual genetic code, looking closely at DNA and RNA sequences, or comparing the molar percentage of certain nucleotides. It's just a way more precise method for figuring out exactly who is related to who on the microbial family tree.
Which brings us to Section 3, the three domains of life. To understand how we organize life, we have to look at the taxonomic hierarchy. Think of this kind of like a cascading address system for living things. It starts at the absolute broadest level, the domain, and funnels all the way down to the highly specific species level. So, for example, a bacteria might belong to the domain bacteria, narrowed down through the order Spirochaetales, into the genus Treponema, all the way to the highly specific species Pallidum. That gives us Treponema pallidum.
The crucial point here is right at the top of that hierarchy. The three ultimate domains are bacteria, archaea, and eukarya. Bacteria are prokaryotes, meaning they lack a true nucleus, but their real defining feature is this rigid cell wall containing a unique substance called peptidoglycan. Archaea are also simple prokaryotes, but they absolutely do not have peptidoglycan. And finally, eukarya. That includes fungi, plants, and us, animals. We have complex cells with true nuclei, and if a eukaryote does happen to have a cell wall, it definitely lacks peptidoglycan.
Section 4, bacteria, the ancient survivors.
Now, even with all our advanced DNA testing today, basic phenotypic shapes are still absolutely fundamental for identifying bacteria.
Most bacteria fall into three classic shapes. You got your spherical cocci, your rod-like bacilli, and your spiral forms. They can exist on their own, or they can form these distinct clusters and chains that act almost like a microscopic fingerprint for identification. And you know, nature always throws a curveball. Back in 1980, scientists actually discovered a bizarre, perfectly square-shaped bacterium. No way, right? But it's true.
And this brilliantly illustrates what makes bacteria so uniquely tough. Remember that peptidoglycan we mentioned a minute ago? Well, here is its chemical structure. It's this incredibly rigid wall that protects the bacteria, and it's built from alternating sugar molecules. You can clearly see the alternating sugars here: N-acetylglucosanane, or NAG, and N-acetylmuramic acid, or NAM. They link together in these long parallel chains via glycosidic bonds. It is basically a microscopic chainmail suit, and it's so substantial that it can account for up to 40% of the bacterium's entire dry weight.
All right, Section 5, Fungi, Viruses, and Beyond.
Because the microscopic world isn't just bacteria, right? There are entities out there that behave very differently. Let's contrast two wildly different groups, fungi and viruses. Fungi are eukaryotes, which actually makes them much closer to us structurally than they are to bacteria. They have thick, rigid cell walls made of chitin, which happens to be the exact same stuff insect exoskeletons are made of. They're mostly terrestrial, and they are the absolute master recyclers of our environment. Viruses, on the other hand, are incredibly, astonishingly small. We're talking 10 to 100 nanometers. You absolutely need an electron microscope to even see them. They're essentially just these tiny packets of genetic material that sit right on the razor's edge of what we even consider to be living.
Speaking of fungi, here is a truly fascinating survival mechanism. Some fungi are what we call dimorphic. This means they can literally change their physical form based entirely on the temperature of their environment. It's basically a superpower. Inside the warm environment of a host's body, say around 35 to 37 degrees Celsius, they grow as round, single-celled yeast. But out in a cooler environment, like normal room temperature around 22 degrees, they completely transform. They turn into fuzzy molds with long branching structures called hyphae. It is just a spectacular biological adaptation.
We've also got some unique microbial groups that kind of bridge our understanding of diversity, like spirochetes and rickettsia. Spirochetes, like that treponema we mentioned earlier, are these highly unique, irregular, spirally curved bacteria. They move in distinct ways and often thrive in environments without any oxygen. Then you have rickettsia, which are a group of incredibly tiny bacteria. They share sizing traits that are almost closer to viruses, which makes them incredibly difficult to study without highly advanced microscopy.
Now, when we put all this classification science to work in the real world, particularly in medicine, we use these exact phenotypic traits to identify specific pathogens on the fly. For instance, if a lab tech sees gram-negative rods with flagella that let them move around, they might very well be looking at Salmonella. If they see clusters of gram-positive spherical cells that are completely still and non-modal, that strongly points to Staphylococcus. And those irregular spirals thriving without oxygen? Yep, that's our treponema. Structuring our knowledge this way literally saves lives by speeding up the diagnosis process.
Finally, Section 6, our microscopic co-inhabitants.
We interact with these microbes constantly, every single second of the day. The text actually outlines 4 distinct forms of cohabitation. The first is symbiosis, which is a cooperative association where both we and the microbe benefit. It's a win-win. Then there's commensalism, where the microbe gets a benefit but it doesn't harm us at all. There is, of course, true parasitism, where the microbe benefits at the strict expense and harm of the host. That's what happens when we get an infection. And finally, saprophytism, where organisms live off dead, decaying matter, recycling life back into the earth. It's so important to remember that we rely on symbiosis and commensalism just as heavily as we fight off parasitism.
Which leaves us with a truly profound realization to end on today. Given that our own human cells are vastly outnumbered by bacterial cells, and considering that microbes have been adapting and surviving on this planet for 3.8 billion years, are we really the hosts steering the ship, or are we just highly complex habitats, wandering through a world utterly dominated by unseen, ancient, and highly structured microbes? It's a fascinating thought to chew on. Thanks so much for joining me for this explainer, and I really hope this changes how you view the invisible universe all around you.